Hello and welcome to PhysioEx exercise number 4, activity number 2. We talked about how PhysioEx is divided into multiple exercises. Each exercise represents a physiological system, and each exercise is subdivided into multiple activities, and each activity is further subdivided into multiple parts. We are finished with activity number 1. So now let's uh, proceed to activity number 2. And like we said, the activity number 2, let's look at so we have finished with part 1. So now we have finished with activity number 1. This is activity number 2, which is titled in the PhysioEx as activity 2, plasma glucose, insulin, and diabetes mellitus. So what we have here in this plasma glucose, insulin, and diabetes mellitus activity, we will do two experiments, which are two parts. So the activity 2 of exercise 4, the endocrine system, is divided into two parts. The first part, we have an independent variable and a dependent variable. And for the second part, that dependent variable is going to be reversed. It will become our independent variable for the second experiment, and we will use it as an independent variable to probe for or find our dependent variable, which is the unknown fasting plasma glucose. So just keep in mind that we will be using a spectrophotometer, and we will be examining optical density. What is optical density? Optical density is how thick and how optically blocking how the solution, how much blockage is the solution providing against the light that is passing through it in the spectrophotometer. So optical density is inversely related to absorbance. So the higher the optical density, the lower the absorbance. So if I have a test tube with 100 milligrams per deciliter if I have 100 milligrams of glucose, then I have another test tube with only 50 milligrams, then obviously the test tube that has 50 milligrams will allow for more light to pass through it. So it will have a lower optical density. It is less dense and it will block less light. But the substance that has more um, um, substance in it, more concentration, that will lead to a lower optical density. So the optical density is inversely related to the absorbance of light, while the optical density is directly related and correlated to the concentration. So the more the concentration, the more the optical density. The more the absorbance, the less the optical density. So in part 1 of the experiment, what we'll do is we will have known samples of glucose, and they will represent my independent variable, and I will put them through a spectrophotometer. And then the spectrophotometer will give me values known as optical density values. So these values, I don't know them yet because I haven't ran my experiment. That's why I labeled them as unknown optical density values. So when I take these test tubes, I have about 5 test tubes with increasing concentration. One test tube is, for example, 5 milligrams per deciliter glucose. The second test tube is about 20 milligrams per deciliter. The third one is 50. The fourth one is 70. The fifth one is 100, for example. So we have increasing um, concentration of glucose. So I do know, it is known for me, I do know how much glucose is in there, but I do not know how much light will pass through it. So I do not know the optical density. I do not know how much light is blocked. So will use the values of glucose that I know, test tubes. I will have test tubes of glucose that I know, and I put it through a spectrophotometer, and then I will try to find out how much the optical density is. Then I will take these values. So now I found out it is no longer a secret for me. It is no longer unknown optical density values. So now I know these values, and I will take them after I generate a plot. I will generate a curve. So I will establish a relationship, a curve from which I can derive an equation. This equation will demonstrate for me the relationship between the data that I plotted on my graph. Then I will take that graph, and on that graph, I will now bring I will now have my known optical density value. So now I know how much 30 milligrams per deciliter represent as optical density. How much optical density will 70 milligrams per deciliter provide for me? And then I will take 5 samples of fasting plasma glucose of patients. So now I don't know. I have 5 patients and I took blood sample from them, but I do not know these patients, the blood sample. I know that they were fasting for 8 hours, so it is fasting plasma glucose, but I don't know how much is it. But I do know how much glucose, how much, because I have a curve, I have a formula. So I will now look in my graph and see the value that these unknown fasting plasma glucose test tubes. So patient number 1, I will take their blood and put it in a spectrophotometer and then look for in my graph that patient's blood glucose is close to test tube number 1 in my previous experiment. And patient number 2, for example, their blood glucose would give me an optical dense value that is closer, for example, to test tube number 3, and so on and so forth. So this will get more clear as we go on with the experiment. So let's go ahead and begin. So our objectives are to understand the use of the terms insulin type 1 diabetes, type 2 diabetes mellitus, and the glucose standard curve. So we have to generate a glucose standard curve. And we will find out how fasting plasma glucose is used to diagnose diabetes mellitus. And we would be understanding the assay that is used to measure this plasma glucose. So let's find out. So this is an introduction, as we said. This is a very important section that is whenever I have an experiment, this experiment, I have to publish it in a journal article with an introduction. This introduction will signify for me how important is the experiment and why should I be running an experiment to investigate the effects of the independent variable on this dependent variable that I'm measuring and so on. So this is just an introduction section talking about some basic information that we already talked about in lecture. So these are the equipments that I will put in my materials and methods. So I talked about the general scheme of my experiment, but obviously we have some materials that I did not mention. Like, for example, the ionized water is used to just adjust and normalize the volume of the fluid through which I will have my light passing inside the spectrophotometer. Then I have a glucose standard with a dropper cap that I will use it to pour and put in known samples of known amount of glucose concentration. And then I have an enzyme color reagent that will later on turn in color, different in color according to how much concentration of glucose. So the more concentration of glucose that I have in a test tube, the darker the color, and the less concentration of glucose, the less the color. And then I have barium hydroxide, which is responsible for reacting and removing all proteins and all substance that may interfere with my experiment, because I'm running the experiment to subject the light onto the glucose. So I do not want light to be interacting with anything else, proteins, plasma, or anything. I need to remove all of that. And then heparin will prevent any blood clots for me in those blood samples. And then I'll have blood samples from 5 patients. and I have these test tubes that I will put in a washer, and then an incubator, and then this is a very important spectrophotometer through which I will be subjecting the light and measuring how much or amount of light is absorbed and transmitted, and then take out a value known as the optical density value to give me a value of how dense this solution through which I projected my light, and I when I put it in the spectrophotometer. So moving on. So we have a pre-lab. Which of the following statements is false? Insulin is a hormone secreted by the beta cells of the endocrine portion of the pancreas. That is true. Insulin promotes the storage of glucose in the form of glycogen. That is true. And it's a hypoglycemic hormone. Then insulin decreases plasma glucose levels, obviously. Insulin is a hormone secreted into the stomach. That is not true. It is an endocrine hormone secreted into the blood to aid in starch digestion. No, that's not true. Now, moving on. Which of the following statements is true? Plasma glucose levels are controlled by the opposing actions of insulin and glucagon. These are antagonists. That is true. Plasma glucose can be converted into glycogen stores in the liver and muscle. That is perfectly true. And glucose is the primary fuel for nerve cell metabolism. All of that is true. A diagnosis of type 1 diabetes mellitus implies that only type 1 nerve cells are responding to insulin. That's not true. And all cells of the body are unresponsive to insulin. That is not true. The pancreas is not producing sufficient insulin. That's true. Usually maybe due to a autoimmune disease where the immune system has amnesia causing autoimmune disease. So you have A and A, autoimmune disease caused by amnesia. So the immune system forgets self from non-self. A person's urine is abnormally deficient in glucose, then that's hypoglycemia. That is not hyperglycemia. That is a low, that is um, the diabetic patient will have glycosuria. That means their urine has a lot of glucose. So if they have deficient glucose, that means they do not have diabetes. Okay, moving on. A diagnosis of type 2 diabetes mellitus implies that only type 2 cells of the body are responding to insulin. The pancreas is not producing sufficient insulin. That's one. The cells of the body are unresponsive to circulating insulin. There is an end organ resistance. But when I say only type 2 cells of the body are responding to insulin, that doesn't make sense. A person's urine is abnormally deficient in glucose. That's not true. Okay, so let's move on. Glucagon is a hormone produced by the alpha cells of the pancreas, not the beta. The poses that is then an antagonist to the actions of insulin. That is perfectly true. Okay, moving on. Submit. Okay, now it's part one. So in part one, we'll be doing this. We will have known samples of glucose as my independent variable, putting it in a spectrophotometer to measure my optical density, which is the dependent variable. And then we will take those values in the next part of the experiment, run them through, and then use that curve. And then I will have unknown glucose, plasma glucose values, and I will use the values that I got here to extrapolate my actual unknown fasting plasma glucose values. So let's begin. So it says in this part of the activity, you will generate a glucose standard curve so that you have points of reference for converting optical density readings into glucose readings measured in milligrams per deciliter. So to generate a glucose standard curve, you will prepare 5 test tubes in ascending order, 30 milligrams per deciliter on the first tube right here, then 60 milligrams per deciliter on the second tube, then 90 milligrams per deciliter on the third tube, then 120 milligrams per deciliter on the fourth tube, then 150 milligrams per deciliter on the fifth tube, and use a spectrophotometer to determine the optical density readings for each of these glucose concentrations. So drag a test tube to the first holder in the incubation unit. Okay, so 4 more test tubes will be put in the incubator. So now we have 5, so we need to add sugar in ascending order. It will tell you So drag the dropper cap of the glucose bottle into the first one. So you see it will have one drop here, meaning 30 milligrams per deciliter, 2 means 60, 3 means 90, and 4 represent 120 milligrams per deciliter, and the last one is 150. So I have increasing amounts of glucose in each tube. Okay, drag the dropper cap of the deionized water. So this is the deionized water, and it will do 3 drops here to just normalize the volume and bring it up to the same level. So that's the role of the deionized water. Then we will mix everything up. After we have mixed everything, then we click centrifuge. So obviously when we're centrifuging, we're spinning against gravity. We will have a supernatant and a pellet. So let's remove the pellet. We have removed the pellet. Then we drag the dropper cap of the enzyme. So now we will have the calorimetric enzyme activity, and the more substance in there, the darker the color, the more substance, the darker the color, the more substance, the less the absorbance, the more substance, meaning the more concentration. The more concentration of glucose, the darker the color of the solution. The more concentration of glucose, the less absorbance. The more concentration of glucose, the less the transmission, and the more the more concentration of glucose means that the more optical density. So the more concentration of glucose, you will have more dense objects preventing the passage. So you will have high absorbance and low transmission. Let's click on incubate, and then we have incubated everything. And so stop and think, why does the color in the tubes vary? The tubes are numbered differently. The tubes contain different volumes. The tubes contain different amounts of glucose. That's the perfect answer. And then select the setup on this. So now we want to put everything into the spectrophotometer. So I've set up my spectrophotometer. Spectrophotometer is ready. So let's drag tube 1. We put it in there. We have dragged tube 1, and then we press analyze. So everything was analyzed. And then that point is the beginning of my curve. So now I knew that I had 30 milligrams per deciliter of glucose in test tube number 1. You see, there are known samples of glucose, but unknown optical density. But now it is no longer unknown because it turned out to be 0.3. So now I've learned, I didn't know how much optical density was 30 milligrams per deciliter, but now I found out. So now let's click on record data. So now I have recorded that in part 1, tube number 1 had this much amount of glucose. That's my independent variable, and that was how much optical density. So now let's do the same thing for tube number 2. Let's analyze that. We analyzed it, and interesting. I knew all along that I had 60 milligrams per deciliter of glucose in my tube number 2, but I never knew what the optical density was. But now I do know it's 0.5. So now let's do the third one, and the fourth, and the fifth. And then let's record the data. Then number 4, analyze, record the data. The last one, 150 milligrams per deciliter. I knew that it was 150 milligrams per deciliter. It was my independent variable. That's why it was on my x-axis. And on my y-axis, I never knew that. I knew that on test tube number 5, I had 150 milligrams per deciliter, but I never knew what the optical density was. But when I ran it through my spectrophotometer, it turned out that 150 milligrams per deciliter comes out to about a whole 1 optical density. So now I'm going to use that curve. So graph the glucose standard curve. This curve, we can extrapolate a formula. Remember y equals mx plus b? You see, for the weather, for example, let's say I want to find out when exactly I will be having a snow blizzard in the upcoming years. This is the year 2024. Let's say in the year 2024, the blizzard was on February 15th. Okay. On the year 2023 was February 10th. On the year 2020, it was February Seventh. So if I go back in time from the year 2000 up to the year 2024, and I record the date of my very first snow blizzard in the winter that led to the stopping of traffic and stopping of school and inclement weather. If I record the years on the on the x-axis, this is my independent variable, and then on the y-axis I plot the date, like I plot when exactly it happened. Then when I put all of these data, I can get a curve. That curve, I can take a formula, make a formula out that curve. Then I can use that formula to extrapolate. In the year 2027, when can I, based on these previous values that I had, when can I expect a snow blizzard? I can even go back in time to 1973 and use the same formula, because it's an entire um, uh, uh, this is a very important graph that I can use the formula to extrapolate future, predict the future, or calculate the past. So this is exactly what I'm doing here. I now know I have a curve right here. So next time, if I have a mysterious test tube, this mysterious test tube represents blood sample fasting plasma blood glucose. This is a blood sample from a patient. I do not know this patient, the blood that the patient has, how much glucose is in there. Is it 30 milligrams per deciliter? Is it 50 milligrams per deciliter? But what I can do is take that test tube with the blood of my patient, put it in the spectrophotometer, run it, run the light through it, and measure how much optical density is that. And then use my optical density. If the optical density came out to be something close to 0.80, then I can tell that this fasting plasma glucose in that mysterious test tube that I got from the patient is almost 100, for example, 120 milligrams per deciliter. So I used my optical density to extrapolate how much the blood sugar of the patient is, because I have already had a curve. So that's what I'll be doing in experiment number 2. Part 2. So now let's do a prediction. To measure the amount of plasma glucose in a patient, the glucose concentration of the sample will be measured. No, I will not be measuring the glucose. I don't know the glucose, but I will be measuring the light, and the optical density will be extrapolated. No, I will not be extrapolating optical density. I extrapolate unknown things. But since I have a spectrophotometer, I will be measuring that. No, that sounds perfect now. The optical density of the sample will be measured, yes, using my spectrophotometer. So I will have a test tube of an unknown or mysterious blood sugar. I know that this test tube has blood in it with an unknown amount of sugar, and I don't know how much is it, but I do have a spectrophotometer, and I will measure the optical density of that sample, and then I will use my curve, and the glucose concentration will be extrapolated from the glucose standard curve. Now that sounds perfect. Okay, now that's part 2. Let's measure the fasting plasma glucose levels. In this part of the activity, you will use the glucose standard curve you generated in part 1 to measure the fasting plasma glucose levels from 5 patients who donated their blood to us to measure, to diagnose the presence or absence of diabetes mellitus. Now, note the addition of 2 reagent bottles. We have barium hydroxide and heparin. Heparin to prevent blood clots, and barium hydroxide to prevent interference from any additional substance, like proteins or plasma membrane or anything else. Because these are blood samples. So the blood sample will coagulate if left unattended, and there are no anticoagulants, and the tissue will start disintegrating. So we need barium hydroxide to react with it and remove all these impurities that would interfere with the passage of my light. So I want the only thing to interfere with the passage of my light inside the spectrophotometer to be my blood glucose. To undergo the fasting plasma glucose test, patient must fast for a minimum of 8 hours. A patient with fasting plasma glucose values greater than or equal to 126 milligrams per deciliter in 2 fasting plasma glucose tests is diagnosed with diabetes. So fasting plasma glucose values between 110 and 126 indicate impairment. So that's pre-diabetes, impairment of insulin-mediated glucose uptake by the cells. Okay? More than that represents actual diabetes. So fasting plasma glucose values less than 110 are considered normal. So we need to know these values. So fasting plasma glucose values less than 110 milligrams per deciliter. Now that's a normal range. From 110 to 126, that's pre-diabetes. And from 126 and above, if you have two measurements of fasting plasma glucose of 126 milligrams per deciliter, that is an immediate diagnosis with diabetes. So now let's begin the experiment. Drag a test tube, put it in the incubator. So now I'll have 5 test tubes. And these are my 5 mysterious blood samples. Drag the dropper cap of the first patient blood sample into the first tube, test tube. So that's 3 drops of blood. Then it's sample number 2, that's patient number 2 into test tube number 2, patient number 3 into test tube number 3, patient number 4 into test tube number 4, patient number 5 into test tube number 5. Drag the dropper cap of the deiodinized water, and that is to bring up my volume, bringing up my volume. Then we'll use our barium hydroxide. Excellent. Then we use the heparin. We click on mix. Then we click on centrifuge. We're going to be removing a pellet, obviously, like we did in the other experiment. So let's remove the pellet. And after we remove the pellet, we drag the dropper cap of the enzyme reagent. So now the darker the color represents more blood glucose. We incubate. And we click on setup. You see, this is a lighter test tube, and this is a darker test tube. Setup. Graph the glucose standard curve. So that is my graph that I will use to extrapolate make a formula that can predict for me the past and that can calculate for me the past or predict for me the future. Okay. Will not be exact, obviously, but it will give us a rough estimation. Okay, so now drag tube number 1. So now let's use tube number 1 to find out how much sugar was in it. So we press analyze, meaning we have the light running through it. So now the spectrophotometer showed me that this yellow line is the optical density of this mysterious test tube. So how much sugar it had, I'm going to make the intersection point representing my. So according to this graph, yeah, the intersection point represents, so now I'm intersecting the optical density on the same graph. So we're utilizing the same graph. So we're intersecting, and that comes out to about 105 milligrams per deciliter. So my optical density is 0.73, and my glucose concentration using my curve happens to be 105. Let's do the same thing for number 2. Analyze. We ran a light through it, and that's how much optical density, which is about 0.79. So let's find the intersection point. That's about the intersection point. 114, 114. And it comes out to about 115 glucose milligram per deciliter. That's for patient number 2. That's sample 2. Now let's look at sample 3. Analyze that. When we analyze that, we will have we recorded data. We've already recorded that sample number 3, optical density 0.89, and this is above 126. This is a diabetic guy. Okay. So now, congratulations, you have completed this experiment. Select submit to record your results. So now I was able to tell patient number 5, whose blood sample was in test tube 5, had 143 as a diabetic patient. Patient number 4 is a diabetic patient. Patient number 3 is a diabetic patient, and 2 is a borderline pre-diabetic, and the patient number 1 is a healthy patient. Okay, so press submit. We submitted everything. Now, post lab, a male patient has had successive fasting plasma glucose readings of 115, 110, 122. He haven't yet hit 126, that mark. Then the healthcare provider will inform the patient that he's borderline, that he appears to have impairment or borderline pre-diabetes, insulin-mediated glucose uptake by his cells. Then, to obtain an accurate spectrophotometric measurement of the glucose concentration in the sample, if you want to obtain an accurate spectrophotometric measurement of the glucose concentration in the sample, water is added to dissolve the glucose. Barium hydroxide is added to prevent blood clots. No, that's the job of heparin. Heparin is added to prevent blood clots. That's excellent. So we could do that. In the spectrophotometric assay used in this experiment, the ... as the glucose concentration in the sample increases. So the optical density increases as the glucose concentration in the sample increases. That is true. A female patient has had successive fasting plasma glucose readings of 130, 140, and 128 milligrams per deciliter. The healthcare provider will inform her that she has developed diabetes. Definitely. She has developed diabetes. To maintain plasma glucose homeostasis, the pancreas will release insulin and glucagon together in order to offset the rise. And no, to offset the rise, we will only secrete insulin. Glucagon-mediated transport of glucose into cells acts as negative feedback when plasma glucose levels rise. Cells will become unresponsive to insulin if plasma glucose levels are chronically low. So glucagon-mediated transport of glucose into the cells. No, that is not done by glucagon. That is done by insulin into the cells. Insulin-mediated transport of glucose into the cells acts as a negative feedback when the plasma glucose levels rise. Excellent. That is perfect. A laboratory technician withdraws a blood sample from a vein in your upper arm, knowing that the plasma glucose concentration is expected to be higher in the arm vein than the index finger. No, it's relatively similar. The plasma glucose concentration will be the same in both. Yeah, we'll do that. Then you have completed the post-lab quiz. We press submit. So now let's answer these very important questions in the review sheet. What is a glucose standard curve? So now we have to define what is a glucose standard curve and why did you need to obtain? Why did you need to obtain one for this experiment? And did you correctly predict how you would measure the amount of plasma glucose in a patient sample using the glucose standard curve? So the glucose standard curve correlates the intensity of the color obtained and measured on a spectrophotometer, the optical density, to the glucose concentration. So the more glucose concentration, the more the intensity of the color obtained and measured on the spectrophotometer. Okay, did you correctly predict how you would measure the amount of plasma glucose in a patient sample using the glucose standard curve? What is a glucose standard curve? It establishes a relationship between the independent variable being glucose concentration and optical density. And then the other part says, did you correctly predict how you would measure the amount of plasma glucose? The optical density of the sample would be measured and the glucose concentration would be extrapolated. Yes, I did. That's exactly what we did. We extrapolated the fasting plasma glucose values based on the intersection with optical density that we knew from part one of the experiment to represent known glucose values. Which patient had glucose readings in the diabetic range? Well, let's see. In the diabetic range, it was patient number 3, that was diabetic, 4, that was diabetic, and um and can we tell if so patient number 3 is diabetic, patient number 4 is diabetic, patient number 5 is diabetic, because it's all um above 126. But you can never tell if it's type 1 or type 2. That can never be told because there is a possibility of autoimmune drug attacking, I'm sorry, autoimmune reaction mounted against the pancreas, um, inhibiting or destroying the beta cells, preventing them from secreting insulin. So patients who are diabetes type 1, they will have no insulin, but patient with diabetes type 2, they will have an insulin, but there is an end organ resistance, um, disabling the cells, the rest of the cells of the body from responding. Okay, describe the diagnosis for patient 3, who was also pregnant at the time of this assay. So patient number 3. Patient number 3 is definitely diabetic, but since she's pregnant, that is also known as gestational diabetes, and it will go usually after pregnancy ends. So that is gestational diabetes and usually resolve after pregnancy ends. Patients 3, 4, 5, and we can't tell if they are type 1 or 2 without further analysis of insulin levels in the blood. If you have diabetes and you have insulin, then that means you're type 2. But if you have diabetes and no insulin, it means you have type 1. I don't mean no as in no, but low, very low insulin. Okay, which patient had normal glucose readings? Which patients had normal glucose reading? Yeah, 105 and 115 sounds about good. So patient number 1 and patient number 2. Patient number 1, patient number 2. And what are some lifestyle choices these patients with normal plasma glucose readings might recommend to the borderline impaired patients? Exercise, healthy diet, and yeah, exercise and healthy diet. We don't want a sedentary lifestyle, and we do not want fast food and high cholesterol food. Okay, so let's press submit. That shows me, you know, all my answers, and then you're gonna select, you're gonna put your name, and then print the friendly version, or maybe save it and send it to my email. Okay. And that should be the end of exercise 4, activity 2. See you in exercise 4, activity 3.